Thanks, Andre, for that really kind introduction, to the Danone Institute for the invitation, and I'm so honored to be in the company that I'm in with Aran and Jens. And they both gave great introduction to a lot of the stuff I'm going to talk about, so I can whip through some of my slides and save a little time. I don't really think I'm an aging researcher either. It's about 6 years ago, I guess, we started thinking about aging in the context of the microbiome. And when we were reading the literature, we kind of observed two major patterns that were out there. What is aging in the gut associated with? Well, one signature seemed to be kind of managing the decline. You want to just put the brakes on bad stuff that's happening as we age. And this is where a lot of the aging field is, kind of trying to slow things down or rejuvenate you back to some prior state. But there was also work showing, that in centenarians or maybe in very healthy older people, there might be differential patterns occurring. And so, for example, in centenarians, there was this observation that there's a steady decline in some of the core taxa that dominate us when we're younger, at least on average, and then a rise of the more subdominant taxa in the gut. And this work was driven by a really talented postdoc from a few years back, Tomasz Wolmański. And the major signature that we found when we were looking at this large wellness cohort that we had access to called Aravel was this thing called uniqueness. We sort of invented this metric, honestly, just by kind of squinting at the data for long enough to kind of explain what it is. If you think about the beta diversity, how similar are two microbiome profiles to one another, how distant are they, or how similar are they? If you project them into some 2D space and every point is a microbiome and distance is sort of how differential the ecology is between the 2 points, your uniqueness score is your nearest neighbor distance in this space. So this red point has a higher nearest neighbor distance than this blue point. It's kind of on the fringe of the cloud. And oddly enough, during the course of the lifespan, it looked like what was going on is you get this steady rise in uniqueness over the decades of life. We didn't quite know exactly what this meant. The Aravel cohort, it was a consumer scientific wellness cohort where people were coached on personalized wellness. And we had access to the data, thousands of people with lots of dense multi-omics. And so this is like a generally healthy set of people. So that's a caveat here to think about. And we validated this in many, many other cohorts. So if you look across the world even, and other groups have done some validation here too, you indeed see this pattern of increasing beta diversity uniqueness as we get older. It can vary a little bit cohort to cohort, but generally we kind of see this happening. And weirdly, it's associated with things that are happening in the bloodstream. And so we kind of correlated this uniqueness score with circulating blood markers in these thousands of individuals. And the major signature we see are these, Jens mentioned proteolytic fermentation of microbes. You see these amino acid fermentation byproducts popping up in the bloodstream. Not necessarily the good guys, right? We often consider these things as bad, like p-creosol or phenylacetyglutamine. These are associated with like heart disease or kidney damage. Yet, they're also super enriched in the bloodstreams of centenarians. And in fact, if you kind of Google phenylacetyglutamine, it's been patented as a biomarker of centenarian status. It's way elevated in the bloodstreams of centenarians, but also in their children's bloodstreams. So it's even a marker of likelihood of achieving a long life. I don't know what's going on. This is still kind of a paradox for us. My suspicion is that when we get a lot older, increasing our protein intake might actually be protective against frailty or muscle loss, whereas maybe in early life, higher protein intake is not so great. I don't know. But getting back to the microbiome, one of the kind of taxonomic signatures of this uniqueness signature is a depletion of gram-negative anaerobes that tend to dominate our guts when we're young, like Bacteroides or Prevotella, depending on where you are in the world, and a rise in a lot of different butyrate-producing Clostridia, which are the kind of blue colors here. So we see a lot of these different genera from this class of butyrate producers that tend to be elevated in people who have higher levels of uniqueness. Kind of collapsing that down back into kind of a beta diversity space where different distance between points is kind of similarity in the ecological structure of your microbiome. We see that uniqueness is clustered up here in the elbow of this curve in these thousands of people. You can paint this by different taxa. So Bacteroides are enriched over in this arm, Prevotella down in this arm, and the crux here is enriched generally in these butyrate-producing Clostridia. You can see a very strong negative association between this uniqueness score and the dominance of Bacteroides in the guts of people. Okay, so then we next looked at a cohort of older folks. So Aravel spanned the whole lifespan, people from 18 years old to 99. We then looked at the Mr. Ross cohort, and this was we had a collaborator who had access to this cohort. It was recruited to look at osteoporosis in men, which was an understudied problem. So it's 1,000 older men between about 75 and 100 years old. And our uniqueness association with age was there, but it was actually weaker than what we saw in the Arabelle cohort. So we thought to ourselves, okay, maybe there is this partitioning going on. Maybe there are differential patterns of aging in the gut. So we found markers of, we found markers of health in this data set. So we could partition people based on their walking speed. So you can walk faster when you're older, you tend to be healthier. Self-reported health, whether you had high or low medication usage, or this life space score, which is a measure of kind of how much you get around in your life. And a composite healthy is you had to be healthy according to at least 3 of these other prior metrics to be composite healthy. But no matter how you sliced or diced it, if you were healthier, you tended to show a stronger association between uniqueness and getting older. And if you showed less good health, that association went away completely. So it seems to be this thing that's happening. There's differential signatures going on here. We also had follow-up data in this Mr. Ross cohort, so we could partition people based on their baseline microbiome composition and ask what happens with survival over a 4-year window. In the Kaplan-Meier here, you can clearly see that if you have higher Bacteroides dominance at your baseline time point, you're more likely to pass away at over 4 years. To control for other covariates, we did Cox proportional hazards, did a lot of different versions of it here, but in the kind of fully multivariate adjusted model, where we're adjusting for age, clinical site, body mass index, self-perceived health, and diagnosis of congestive heart failure, so trying to account for other factors that could be related to mortality. Controlling for those, if we look at people who've achieved very old age, 85 plus, we see very significant associations. So if you have low uniqueness or high Bacteroides, you're more likely to die over a 4-year window. So, it's correlated with health in older age, and it's also predictive of survival. Whether this is a causal signature or this is a survival effect is TBD, right? Like, the older people might just be enriched for these signatures because they happen to survive. So we're still trying to understand what exactly is going on. We wrote a little perspective piece after this paper, kind of pushing back against some of the thinking in the field. So should we rejuvenate the gut, right? Should we give FMTs from 20-year-olds to 80-year-olds? Maybe not, is our take home. Because a lot of folks, when we're young, have this Bacteroides-dominated microbiota, and perhaps if we start transplanting that into very older folks, that might not be so good. And maybe it's just fine in a 20-year-old, but in an older person, it might actually do some damage. It seems that a decline in the dominance of these gram-negative anaerobes and the rise in these butyrate-producing Clostridia is associated with better outcomes later in life. We don't know what the mechanisms are driving these patterns. I'm sure it's diet. I would guess diet and lifestyle. And then how do we target? What is the outcome variable we want to target here? I don't want to live to 100 years old and be decrepit for the last 40 years of my life, right? I want health span, not lifespan. So let's think about what are metrics we want to improve with interventions that are focused on healthy aging. So then thinking about that, we moved to frailty metrics. So a really cool colleague joined my institute, Dr. Alice Kane. She was did a postdoc at Harvard in aging, and then came to ISB, and she looked at frailty in mice and in humans. And in this human study, she found that if you look at survival outcomes in a human population, frailty does a whole lot better at predicting survival than other things like metabolic syndrome, like chronic diseases, essentially. So it seems to be a very useful marker of outcomes, of health, and of survival. So we have the ability to now calculate these scores in the Arabelle cohort that I talked about earlier. So we can do this either through looking at clinical chemistries, so things like LDL cholesterol, A1C. We can aggregate a bunch of these things and calculate a score. We can also use self-reported questionnaire data about health and create a score there, or do a merged score. We tend to now use the merged score because it seems to be doing a little bit better job. Alex Carr in the group started running regressions on the microbiome data versus frailty. If you control for age, sex, BMI, and the first 4 principal components of genetic ancestry, this is the kind of stuff we see, which is kind of what we were expecting to see. So you pretty much get a lot of butyrate producers that are negatively associated with frailty, independent of age and these other covariates, and then a bunch of kind of a rogues gallery of bad guys that are positively associated. About 15% of the variance in frailty was explained by the microbiome after adjusting for these other covariates. So it's not nothing here. There's a fair amount of the variability in our healthy aging phenotype that seems to be explained by the microbiome. This also validates in other data sets. So this is a recent study in Nature Aging from another group in China, and they found the same exact things associating with frailty. It was essentially positive beneficial effects of butyrate producers and negative associations with a lot of the gram negatives, like the Bacteroidetes and the proteobacteria. So butyrate, I've been saying butyrate a lot. What is butyrate? It's a short-chain fatty acid. Jens brought this up before as well. It's probably the most potent of these short-chain fatty acids that the microbiota produce. It's super important. The only way we get it is through our microbes, and we're really underexposed to it in the developed world because we don't need as much fiber as we used to, right? We're lucky to get 10 grams of fiber in the US a day, whereas maybe we got 50 to 100 grams historically. So we're underexposed to this thing. What does it do? Lots of stuff, but maybe I'll point you towards one of its main functions that we know about is it's an HDAC inhibitor. And so, for example, it affects T cell differentiation. It biases T cells towards T regulatory cells rather than Th17 cells. And it tends to lower systemic inflammation across the body and help to normalize metabolism as well, like blood glucose and so on. So we want to make more of it. But it's hard to predict butyrate production from a given person's microbiome because whether you produce a lot of butyrate isn't simply a function of how much Faecalibacterium presnitzii you have in your gut. It's a coincidence of both your dietary intake and the complex cross-feeding interactions that are happening in your microbiota. Our group has done some thinking about how to do this integration and do these predictions. We built a tool called MyCom. Christian Diener, a very talented research scientist in the group who's now faculty in Austria, it essentially is a tool that allows for flux balance analysis, genome scale metabolic modeling of complex multi-species communities that consist of hundreds of taxa. You use sequencing data to constrain which organisms are in the system. You pull down curated genome scale metabolic models from those organisms to make a kind of simulacrum of their gut metabolism, and then you constrain these models with dietary intake. There's a lot of ways to do that. We strip the diet of components that are consumed by the host, so things that are absorbed in the small intestine by using the Recon 3D human metabolic reconstruction, and then add a few things like mucin and bile acids to the milieu. And then we can predict growth rates of all the bugs in the system and the kind of fluxes of net consumption, net production, and the cross-feeding of metabolites in the system. We call these things microbial community scale metabolic models, or MCMMs. Okay, so we did a little bit of validation of these predictions in a prior paper by Nick Bowman. I'll just show you one piece of that analysis. We had data from Arabelle again with blood clinical chemistries, about 130 different things you can get measured at the doctor, and predictions of butyrate flux from their guts, making a very dumb assumption that they're all eating the standard American diet, which they're probably not all eating that, but close enough. And when we do this, we find that there's this many metabolites, 20-some metabolites that survive FDR correction, and they make sense with our intuition. So a lot of these are markers of lipid health. So if you are predicted to produce more butyrate in your gut, you tend to have lower LDL cholesterol, lower triglycerides, you have better insulin sensitivity, and you have lower systemic inflammation in general. So we think that it's telling us something resonant or useful or real biologically. What if we apply this to this uniqueness metric we were looking at before? Well, this is all kind of new work now that I'm going to show that hasn't been published yet. But when we run these models on the Arabelle population and look at uniqueness versus predicted butyrate, they're pretty well aligned actually. R of 0.53, not perfect, but it's pretty strongly associated. If you have higher levels of uniqueness, you are predicted to produce more butyrate, assuming a standard American diet. And this, aligns with this idea that the Clostridia are increasing as well in this group. So now I'm going to take another kind of hard pivot here towards statins, and this does actually make sense, and it relates to what I said before, and I'll try to explain. So we had another paper by Tom looking at how the microbiome's composition can affect our responsiveness to statins, this drug that lowers cholesterol in the body. And we looked at what are called enterotypes. This is a method of coarse graining or clustering microbiomes into groupings. We did this because another group had used this method on a statin study, and we had to kind of replicate some of their results. But it ended up being kind of useful. So this is, again, the Arabelle PCOA that I've shown you before, this kind of V-shaped thing with this elbow and two arms. And you get 4 groups reproducibly. You see this in Europe and the US especially. There's a Prevotella group that's dominated in the genus Prevotella. You have this Ruminococcaceae group that's dominated by gram-positive anaerobes. And then you have 2 groups dominated by Bacteroides, BAC1 and BAC2. What differentiates them is this BAC2 group is very much depleted in Faecalibacterium, which is this dominant butyrate-producing microbe in the gut. And if we link this back to uniqueness, it's sort of, it's inverted, this plot, but it's the same people, essentially. And you see that it's the high uniqueness folks here that are in this Ruminococcaceae cluster, essentially. Okay, so if we look at so poor metabolic health and obesity, I forgot to mention, has been associated with this BAC2 enterotype. If we look at the statin responsiveness, just to give you a summary of what that paper found, is if you look across these enterotypes, we find that the on-target effect of the statin is maximized in the BAC2 group. So if you're in this enterotype, you tend to have the strongest on-target effect of statins. This HMG molecule, which is the upstream molecule, the enzyme that's being inhibited is HMG-CoA reductase, and HMG builds up if that enzyme's being inhibited. In the Ruminococcaceae group, you still see a significant uptick in statin users, but it's not quite as much. And here's the LDL lowering here. But one of the side effects that's underappreciated about statins is they actually kind of somewhat increase your risk of type 2 diabetes and perturb your insulin sensitivity. And it's the same BAC2 group that shows the biggest perturbation to their insulin resistance when they take statins. So they're most vulnerable to the side effect, but they also get the biggest on-target effect. Okay, so going back to frailty and the Arabelle cohort, if we group people into these enterotypes again, we find that it's this BAC2 group that shows the highest average frailty values, and it's quite significant. And then concordantly, this Ruminococcaceae group, this gram-positive anaerobe-dominated group, it shows on average the lowest frailty values in this cohort. And this is a very big mix of people. It's people from 18 all the way up to 90 something. So it's very heterogeneous, but the patterns are quite significant. If we project this back into our uniqueness scores and our predicted butyrate, it aligns well with our expectations. The Ruminococcaceae group shows this highest level of uniqueness on average. BAC2 is fairly low. And then predicted butyrate, as you might expect, Ruminococcaceae is highest, and then the BAC2 group is lowest. And we're stratifying by sex as well to kind of show you how that falls out. And this is work, I should say, by Alex Carr in the lab, postdoc. So one unique thing about the Arabelle cohort is it's longitudinal. They actually had an intervention, so people were given a healthy lifestyle intervention. They were coached to improve their diet. That was mostly just being told to eat more fruits and veggies, and they were coached to exercise more. And so across this population, they were all called once a week by their coaches and pushed to do all this. And what's strange about this cohort, if you look at most human cohorts and do frailty metrics, you find that on average we increase in frailty as we get older. In Arabelle, over a 6 to 12-month window, most people went down in their frailty. So the dashed line is your 0 line, normalized to yourself at baseline. So most people are improving in their frailty over time. But some of these enterotypes can do better than others. So if you're BAC2 and you're a male, you can really bring down your frailty. These are the folks that probably had the worst lifestyles to begin with. This enterotype, like I was mentioning, is associated with obesity, diabetes, metabolic syndrome. So if you improve your diet and you already have that lifestyle, maybe you just have more to gain from having a lifestyle intervention. And the microbiome allows you then to target people for these interventions. For women, it was Prevotella, the Prevotella enterotype that showed the biggest drop in frailty. We're still working on this analysis, but we do see mediation. We see some interesting hits. We often are seeing sex hormones pop up in a lot of different human phenotypes, and this is true here as well for aging. So if we look at your enterotype status as the treatment, which is a little weird, I know, for mediation, and then your blood, this hormone, what is it called, cortisone glucuronide, as the mediator on frailty, we do see a significant mediation. About 21% of the effect of the enterotype on frailty is seemingly mediated via the reduction in this hormone that's a derivative of cortisol in the Rumina-Caucasia group. This all validates in the Mr. Oz cohort. So if you look at a different human cohort, we also see this association with frailty. And if we change our Kaplan-Meier, instead of looking at Bacteroides dominance, but just look at the two enterotype groups, we see kind of the same thing we would expect. If you're enriched in the gram positives, you tend to survive. In enriched in the gram negatives, you tend to not as well. So the take-home points. I would plead with you, don't give fecal transplants from 20-year-olds to 80-year-olds. It's not like the blood boy thing that the billionaires are trying to do. Probably not a good idea. There probably are some 20-year-olds who are perfectly fine donors for an 80-year-old, but you got to find which one. There is no average microbiome, right? The average human has one testicle and one ovary. You know, the average microbiome isn't a thing. I think we want to target these interventions. It looks like having higher dominance of these gram-negative anaerobes is probably not great for your health span. Higher levels of the gram-positive seems to be better. I'm pretty sure that diet and lifestyle are the things that are driving all of this, and we don't quite know what's going on yet. If anybody's out there reviewing grants for the NIH, we put one in, and it's going to be reviewed in July. We would love to dive into this. Like Iran, we have some methods for trying to infer diet from metagenomic data, and we want to try to see if we can learn out what are the drivers of these effects. And with that, thanks to the trainees who did all this work, the funders, and to all of you for listening.
Thank you, Sean. We have time for a few questions. Yes.
Yeah, thank you for this very interesting talk. I am curious about the arrival cohort and the intervention when you have a different frailty difference for some enterotypes. Do you have the enterotypes data after the intervention to see if participants changed?
That's a great question. We maximized our N by only using baseline microbiomes. So we have, for about half of the people, we have follow-up microbiome data. And we will look at this, yes. But I wonder, right? So what I suspect is going on is that having this BAC2 enterotype, you're just more likely to die. And especially when you get into your 80s and 90s, you just stop seeing those people in the population. So it's not that like the microbiome is evolving and changing per se, it's just a survival effect. But if we do see that you can transition from BAC2 to one of these other enterotypes, maybe there's hope for those of us who have another enterotype that we can get to the right track.
Thank you.
Claire?
Hi, Sean. That was great. Some of the latest things that I find fascinating from aging research is that it's not really a linear decline, but it happens where it appears to happen in physiological distinct bumps or phases, with the first one in the mid-40s, then the next one in the 60s, et cetera. Most of the stuff that I've seen linking microbiome with aging may not take this physiology into account. They may model age linearly. Have you looked at the possible associations between decline or changes in the microbiome during these specific phases of aging?
Yeah, I mean, this is something we called out in our original paper, because if you look at the uniqueness over the decades, it's pretty flat until you get to about 40 or 50, and then it starts to go up. And so that's like the first inflection point you were talking about. And then it's not a linear increase. It's like an exponential increase as you progress through the decades after that. So I think it does align very well with the immunology aging data.
Yes.
Sasha Zharnakova-Groningen, great talk, thank you. I'm curious about this uniqueness parameter. It's known and very well shown that other omics layers show the same, particular methylation, but also proteomics and even gene expression. I was curious if you ever compared whether microbiome uniqueness goes along with uniqueness in other biological measurements? Do people become unique in all aspects, or is kind of microbiome separated from other omics?
That's a great question. It's like this entropy theory of aging, right? Are we just kind of like losing regulation and drifting as we get older? And I think what's going on in the gut, it's more of a factor that we harbor a lot of different disparate species and genera from the gram-positive group, and we have like a fewer set from the gram-negative group. And then that, as the gram-negatives are declining, that drives this increase in uniqueness. So I think there's more coherence, I will say, to the microbiome signature, whereas in the methylation, it's just kind of a loss of regulation.
Or maybe a lot of effects.
Yeah.
As well from microbiome. I think it's very interesting.
Yeah.
Thank you.
Good question.
We'll take one last question.
Hi, really interesting talk. Thank you so much. I was really interested that you saw a difference in the enterotype that was driving the improvement in frailty between the genders. I was just wondering if you have any theories about this, or have you looked a little bit more into why that could be?
It's a mystery. So Alex Kane, who I called out, she specializes in sex differences with aging, and it's really interesting, right? In humans, women have higher frailty, but they live longer lives. Men drop dead earlier, but have lower frailty. And how do you square that circle? We see both in mice and in humans these really variable effects. We have one mouse study that I didn't talk about where we were using NAD supplements, which are very controversial now because maybe they aren't going down with age, or they don't look like they are actually. But when we supplement, we don't really see much direct effect of those supplements on host physiology, but we see an effect on the microbiome, and it's an increase in Anerotruncus colihominis, which is a butyrate producer that is enriched in centenarians, or it has been shown to be in a couple of papers. And it has an effect, it mediates lower frailty in one sex, but not the other in mice. And so the microbiome effects in sex seem to be intersecting, and they have variable effects, and I don't think we understand why yet, but I think it's really fascinating.
Okay, well, cool stuff. Thank you so much.
Great, thank you.